Dear students, welcome to this course of basic genetics. Myself Richa Arya, I am from Department of Zoology, Banaras Hindu University. We are currently discussing this unit 1 and week 1, and today we have the second class on elements of heredity and variation. So, in our previous class we have discussed the historical aspects that were already known to Mendel, that was known to Mendel, or maybe people who are working at the time of Mendel like Galton and Darwin. And we also discussed about some basic things that Mendel chosen for his experimental setup, like pea plants and different characters and trait. We have already discussed that at length, and so today, as I mentioned in my last class, we are going to focus more on the Mendel's methodology. So, Mendel's used true breeding plant and hybridization for the crosses. I will talk about it in a minute, but just wanted you to list these facts that Mendel was using for designing his experiment. He was very meticulously collecting his data in a quantitative manner, like how many flowers, how many seeds, shape of seed. So, he was very discreetly collecting individual data sets so that he can analyze it statistically. Right. So, those were the methodologies Mendel employed, and so as I said just in a previous slide, I mentioned that he was using true breeding plant, and I also mentioned in my previous class that there are several terminologies that Mendel used at his time, and maybe in current times they may be changed, but we should be aware of what those terms are and in what context they were used. So, this true breeding, or now we many times say pure lines, pure breeds, what exactly they are? So, they are the plants or animal which if they self breed, if you self breed them, they will give you the same genotype. We will come to the genotype and phenotype in a minute, but understand this concept that the true breeders always give you the same phenotype over and over again. There will be no variability, and we are getting the same phenotype because they have the same genotype. There is no mixing, no outside mixing of different characters. So, that is one important thing. And how exactly that was happening in case of pea? Because I already mentioned that the pea plant is having this bisexual flower where male like stamens and the female part, they are together, right. So, in that flower itself the stamen can fertilize and then make the fruit. So, that is one of the strength, and that is when this kind of phenomena happens that creates the true breeding lines over the time, right. So, and this is the way to understand if a line is true breeding or not. If you self fertilize it, then you will get the same phenotype, that the parents were having, right. So, now the hybridization, the second thing that I was saying. So, what Mendel was doing, he was crossing different plant characters. So, you can see he was crossing tall plant here with the dwarf plant here, right. So, that was the hybridization he was doing, right. So, when he was hybridizing, you can see I have also written in front of T the tall plant capital T 2 times, right. So, that is its genotype. So, what is phenotype? Phenotype is tallness. Phenotype means what we are seeing with eyes, and genotype is the genetic constituent of that plant, right. So, this plant is tall because it has 2 copies of a gene, and because of the 2 homologous chromosomes, and this is denoted as capital T, capital T. For tallness, capital T is the genotype, and for dwarf plant small t is the genotype, right. So, that is how we are discriminating between different traits, right, and when we are putting them together, that is when we are saying they are homozygous, and we will talk about what heterozygotes are in a minute, right. So, this is important to understand. So, now let us come straight to the law of segregation. That was the first law, and this law was proposed by Mendel. As I just said, that Mendel postulated, we will talk about it, and then later 3 scientists, when they independently discovered Mendel's work, they understood those postulates and created these law. So, the first law of Mendel is law of segregation. So, each individual carries 2 allele or Mendel's trait separate during gamete formation. That is what we learned from this law. So, law of segregation, what it says, that when you have a parent which is tall and genotype is capital T, and you cross this tall plant with a dwarf plant with a genotype a small t, in the first generation you will get the tall plant, right. So, a tall plant when crossed with a dwarf plant, the first generation we get the tall plant. So, now another few more terms. So, you can see P, P1, P2, that is how Mendels have written. So, P stands for parents, right, and F, you will see F1, F2, F3, that is the first filial generation, second filial generation. So, F stands for filial, right. So, this is the first filial generation. When the genes of tall and dwarf plant come together, we only see the tall plant, no dwarf plant, right. So, initially people were thinking that the trait was disappeared, maybe blending possibly is happening in these cases. But what Mendel was able to identify is that when Mendel self crossed these tall plants, this is a tall plant, this is capital T, small t, right, because they will talk about it later why we are saying capital T, small t. So, this when they are self crossed, then what is happening is that these progeny of these cross in F2 generation, what we are seeing, tall plant, fine, I mean they were tall in F1, so if they are tall in F2, that is not a problem. But what we are seeing here also is a dwarf plant, right. So, which means that there is no blending, the traits maintain their integrity. In next generation, although in first generation they disappeared, but in next generation they come back. That is great observation. And what Mendel did, as I said, that he was meticulously doing thing, he was counting numbers. You see, he counted how many tall plants he was having, 787, and how many dwarf plant he was having, 277, and so the ratio was about 3:1 for tall to dwarf. And because we are talking about phenotype, this is the phenotypic ratio. So, in monohybrid cross, monohybrid means when they are taking only one character at a time, like here it is the tallness. If you take the flower color, fine, if you take the seed shape, fine, but one character at a time. So, that is monohybrid cross, and in a monohybrid cross you find the phenotypic ratio as 3:1. And now look at this this thing from a genotypic point of view, right. So, from a genotypic point of view, what I already informed you, the tall we are writing capital T and dwarf small t. So, again when a tall plant is making gamete, then what are the possibility of what types of gamete it could make? It could only make two type of gamete, and both the type are same because they are capital T and capital T. So, all the gamete that a tall plant is making are capital T. Same is true for dwarf plant. All the gametes that it is making are small t. And so, the tall plant in the F1 generation is heterozygous. So, what I said, they were homozygous and here it is heterozygous. So, heterozygous means when it has one trait of one type and another of another type in a character. Under a character, two alleles of a same gene are different. One is capital T, which is bringing tallness, and another is a small t, which is bringing dwarfness, right. So, when you cross this plant by selfing, right, self crossing means same genotype with same genotype, then what kind of gamete it will make? See here both the gametes are same type, but here one gamete will be capital T and another type will be small t. Same is true here. So, what exactly is happening? A possibility of making different type of gamete. If I reframe my sentence, then in case of homozygous plants like here, 100% of gametes are of same type, but here there are 50-50 chances that you get capital T or you get small t. So, that is an important thing that we should understand when we are talking about homozygous and heterozygous, right. So, when he crossed them, then in F2 generation what he found? He found that there were tall plants, right. So, if you make these combinations like that, right, then you come up with all different sorts of combinations. So, what we are going to get? Capital T together, capital T, capital T small t, and then only small t, small t. So, that was the constituent in the F2 generation. And what was the genotypic ratio? You can clearly see capital T 1, capital T and small t together are 2, and then small t 1. So, the genotypic ratio was 1:2:1. And to understand this thing, the genotypes, what are the predicted genotype and how many will come, Punnett has made really excellent way to look at it. What you do, you plot the male gamete on one axis and female gametes on another, and then you keep on putting them together, and that is how you can also count. So, in the monohybrid cross it is easy, you do not need really to make the Punnett square, but when we will come to the crosses where more than one characters are involved, you will see that how helpful the Punnett square will be. So, the Punnett name, as I have already written, it is the name of its inventor, and this square is known by his name. So, outcome of Mendel's all the experiment that he has done using single characters, he proposed these 3 postulates, right. So, totally he proposed 4 postulates. Out of 4, 3 belongs here. So, what he found? He found that unit factors are in pair. So, each trait is controlled by 2 unit factor, 2 allele, we just talked in a minute ago. So, these unit factors in pair means each trait is controlled by 2 unit factors, and 2 unit factors means 2 allele. So, they are inherited from the parents. And then he also talked about dominance and recessive. So, I want you to clarify, there are only 2 laws, but and in those 2 laws, law of dominance, there is no law of dominance. Dominant and recessiveness is part of Mendel's postulate, right. So, because in F1 generation he saw only one trait shows up, but another one is hidden. So, whatever shows up he called dominant, and the one which we are not seeing is recessive, right. And in F2 he identifies that even the recessive trait show up back again, although it was hidden in F1 generation, but in F2 generation you start seeing both the trait in specific ratios, right. So, during gamete formation the 2 alleles of the trait separate, to each gamete carries only 1 allele. So, those were the first 3 postulates of Mendel which form the first law of Mendel, law of segregation. Now comes to the law of independent assortment. Remember, in independent assortment what is the difference? The difference is that he was using 2 characters, before 1 character, now he is using 2 characters, right. And then what he is taking genes of different traits, like you can use them in different combinations and they are assorting independently. So, this rule actually is having some exception, we will talk later in later classes, but for this class just see what Mendel did and how independently his characters were segregating from one generation to another. So, to do this he used a dihybrid cross. Now, it is clear to you, monohybrid when he is using only one trait, and dihybrid when he is using 2 traits, maybe trihybrid when he is using 3 characters, right. So, 1 character, 2 character, 3 characters are mono, di and trihybrid respectively. So, here I am going to show you with this example of dihybrid cross where we are taking these 2 characters. What are the character? Round and yellow and wrinkled and green. These are the seed of P, and he has taken one class where the seed were round and in another class they are wrinkled. So, those are the 2 traits of the seed shape, right, and the other one is seed color. Color is in one case yellow and in another case green, right. So, you can see what we have written. So, to depict roundness we are saying capital R, to say yellow we are saying capital Y, and for wrinkle small r, and for green small y. In F1 generation we are seeing heterozygote. I hope that now you are very much familiar with this homozygous, heterozygous. So, these are the pure lines, right, and then this one is the heterozygous. So, when he self-crossed these F1 progeny, what he identified? He identified that all sort of possible combinations were present in the F2 progeny. He found round and yellow, round and green, yellow and green, wrinkle and green, right. So, all combination of shape and color, they are present here. But again, this I am emphasizing over and over again that counting numbers created the difference. So, you see, although every category is present, but they are present in different numbers. The numbers are listed, written below, and these numbers are in a specific ratio. So, they are in a ratio of 9:3:3:1, right. So, they are present, but not every genotype or phenotype is present in equal ratio, remember that. And again, as I showed you before, to make the Punnett square, you can understand the genotype, and now you can understand why it is important. So, you see, male gametes are on one side. So, how we are making gamete? So, in a gamete you will get one copy of a gene, like one allele, right. So, you will get capital R, capital Y, or capital R with a small y, or a small r with capital Y, or small r with a small y, right. So, this is what you will get different combination from the male gamete, right, in F1. So, this Punnett square we are making from F1, right, when we are crossing, self crossing, that is when we are talking about the possibilities. And same possibilities are, because it is a self cross, so same possibilities are for female. And by combining these genotype, we can see how this 9:3:3:1 ratio fits well with the number actually that he was counted, right. So, this is something some important point to understand. One more thing, just by looking at the seed, you do not know the phenotype, right. So, a round and yellow could be this, right, capital R, capital Y, small y, or round and yellow could be capital R, capital Y, capital Y. So, how we will know the genotype? So, to understand the genotype, what we apply test cross. So, in a test cross, remember, whenever we have a F1 progeny or an organism and we cross back to its recessive parent, remember, recessive is the important word. So, to do test cross, we always cross the offsprings with their recessive parent, and because recessive parent will not show up, but then whatever will be present in the parent you will start seeing. If you cross it with the dominant parent, then the dominant will come, you will never see the recessive if it is present in the offspring. So, there are test cross and back cross. Back cross is something when we cross the offspring with any of the parent, whereas test cross specifically when we are crossing them with recessive parent. So, in the test cross, when you analyze the F1, then you will see all the possible combinations will separate in a ratio of 1:1:1:1. So, no one is affecting each other, otherwise it will never come to 1:1:1 that you will see in our later units, where we will be talking about extension of Mendelism, where gene interacts with each other and then they are linked with each other and they affect the passing of one character from one generation to another. That is for the later concept, but here when the genes are independent of each other, they do not influence each other, you will always see a ratio of 1:1:1:1. So, here comes the Mendel's fourth postulate, which is independent assortment. Remember, independent assortment is always when we are talking about more than one characters. See, seed shape and seed color. So, when there are more than two characters, then we are saying that the two characters are independently assorting, and that is what he learned. And I already said that we will be discussing the extension of Mendelism in our later units. So, now I am just going to revise the whole concept. So, total Mendel have proposed 4 postulates. I have already discussed and they are listed here in the slide. And Mendel, all his work that he has done very meticulously over the 8 years, he published in 1865. He presented in the Society of Brno where he was working. Although not many people, even as I said, Darwin and Galton, who was his contemporary, were not aware of Mendel's work, because probably it was a small place and they did not get a chance to know his work as well. And again, it is very important, as I emphasized in my previous class and in this class too. There are several terms that Mendel used and we are using them in different ways today, like genetic characters, right. So, characters are like height, color. Traits, we are talking when Mendel say trait, we are talking about alleles, like the variation, like in case of height short and off, in case of color character round and yellow and green, right. And then methods of inheritance, like how they are getting inherited. They are dominant trait and recessive trait. So, in F1 generation, how we are knowing who is dominant, who is recessive? When we are looking at F1, in F1 whatever the character is being shown is showed up, we are calling that as dominant trait, and which is not being seen, we are calling that as a recessive trait, right. And then breeding. So, true breeding and hybrid. True breeding I already told you, those are pure, those are homozygous, they always give you the same. Whenever you cross them by selfing, you self cross them, you will always get the same genotype, whereas hybrid will always show you, a phenotype where some, though they were tall, but then you see a dwarf segregating in a specific proportion. So, they are hybrid or heterozygous, right. And then he talk about phenotype and genotype. Phenotype that we can see with eye, and genotypes are the genetic constituent of the plant, right. And then the genetic, he used the word factor. Now, we know the factors as genes, and then he proposed from his work the two laws have been proposed, law of segregation and law of independent assortment. Remember, no law of dominance. So, well, as I said, Mendel died very early and at the age of 61, and after his death, at least 35 years after his death, his work was rediscovered in 1900 by Hugo de Vries, Carl Carolus and Erich von Teschmark. So, these scientists, they were independently working, and when they were working on their own problem, they also discovered that Mendel long back has also worked on similar problem, and they started restudying his work. And so, with that we are coming towards an end. We have already 2 classes on this topic, and I want you to do some activities so that I can understand how well you are following the concept. So, with that now we are going to talk about a little activity that I want you all to do. So, here are the questions that I want you to look at and work. So, your first activity will be to write the genotypic ratio in the dihybrid cross that in the example that I have discussed. So, in this example here, I have written the phenotypic ratio, but not the genotypic ratio. So, I request you to look at the problem and then identify what are the genotypic ratios in this particular cross. And second thing that I want you to do is, I want you to take 2 of your own characters, right, like flower color or seed shape together or whatever you like, and then draw upon it a square to predict the offspring ratio. Identify the phenotypic and genotypic ratios for F2 generation. So, you already have seen with the round and yellow and green and wrinkled, identify any 2 different characters and then do this activity. And I also want you to think, how would Mendel's law change if he worked on polygenic traits. So, polygenic traits are where not just one gene is regulating a character, right. So, that is what the strength of Mendel, and that is why we know so much about genetics today. What if he would have started with polygenic, and he will be entirely confused. But I would still ask you to think more would happen if these traits, any of them, just assume that they are polygenic, and then what could be the outcome and how it could have changed the results that Mendel have. So, with that I thank you all for your time and attention, and I hope that these classes are making some difference in your basic understanding of genetics, and I will be available to help you understand the concept better. And if you have any questions, you can always contact me at my email, or you can post the questions. Thank you.